Our epidemics of dietary disease have prompted a great deal of research into what humans are meant to eat for optimal health. In 1985, an influential article was published proposing that our chronic diseases stem from a disconnect between what our bodies evolved eating during the Stone Age, during the last 2 million years, and what we're stuffing our face with today, advocating for a return towards a hunter-gatherer type diet of lean meat, fruits, vegetables, and nuts. Though it may be reasonable to assume our nutritional requirements were established in the prehistoric past, the question of which prehistoric past remains. Why just the last 2 million? We've been evolving for 25 million years since our common great ape ancestor, during which our nutrient requirements and digestive physiology were set down, and therefore probably little affected by our hunter-gatherer days at the tail end. So what were we eating for the first 90% of our evolution? What the rest of the great apes ended up eating, 95+% plants. This may explain why we're so susceptible to heart disease. For most of human evolution, cholesterol may have been virtually absent from the diet. No bacon, butter, trans fats, and massive amounts of fiber, which pulls cholesterol from the body. Now this could have been a problem, since our body needs a certain amount of cholesterol, so our bodies didn't just evolve to make cholesterol, but preserve it, recycle it. Our bodies evolved to hold on to cholesterol. So if you think of the human body as a cholesterol-conserving machine, and plop it into the modern world of bacon, eggs, cheese, chicken, pork, and pastry, well then no wonder artery-clogging heart disease is our #1 cause of death. What used to be so adaptive for 90% of our evolution, holding on to cholesterol at all costs since we ain't getting much of it in our diet, is today maladaptive, a liability leading to the clogging of our arteries. It seems our bodies just can't handle it. As the editor-in-chief of the American Journal of Cardiology noted 25 years ago, no matter how much fat and cholesterol carnivores eat, they do not develop atherosclerosis. You can feed a dog 500 eggs worth of cholesterol and a stick of butter, and they just wag their tail. Their bodies, evolved from wolves, are used to getting, eating, and getting rid of excess cholesterol, whereas within months, a fraction of that cholesterol can start clogging the arteries of animals adapted to eating a more plant-based diet. Even if our bodies were designed through natural selection to eat mostly fruit, greens, and seeds for 90% of our evolution, why didn't we better adapt to meat eating in the last 10% during the Paleolithic? We've had nearly 2 million years to get used to all that extra saturated fat and cholesterol. If a lifetime of eating like that clogs up nearly everyone's arteries, why didn't the genes of those that got heart attacks die off and get replaced by those that could live to a ripe old age with clean arteries, regardless of what they ate? Because most didn't survive into old age, didn't live long enough to get heart attacks. When the average life expectancy is 25, then the genes that get passed on are those that can just get us to reproductive age by any means necessary, and that means not dying of starvation. So the higher the calorie foods, the better. So eating lots of bone marrow and brains, human or otherwise, would be a selective advantage, as would discovering a time machine stash of Twinkies, for that matter. If we just have to live long enough to get our kids to puberty to pass along our genes, then we don't have to evolve any protections against the ravages of chronic disease. To find a population nearly free of chronic disease in old age, we don't have to go back 1 million years. In the 20th century, networks of missionary hospitals in rural Africa found coronary artery disease virtually absent, and not just heart disease, but high blood pressure, stroke, diabetes, common cancers, and on down the list. In a sense, these populations in rural China and Africa were eating the type of diet we've been eating for 90% of our last 20 or so million years, a diet almost exclusively of plant foods. How do we know it was their diet that protected them and not something else? Well, in the 25-year update to their original paleo paper, they tried to clarify, look, they did not then and do not now propose that people adopt a particular diet just based on what our ancient ancestors ate. Dietary recommendations must be put to the test. That's why the pioneering research of Pritikin, Ornish, and Esselstyn is so important, showing that plant-based diets can not only stop heart disease, but have been proven to reverse it in the majority of patients. Indeed, it's the only diet that ever has, perhaps because that's what we ate throughout the vast majority of our evolution. Studies dating back nearly a century noted a striking finding. If you take young, healthy people, split them up into two groups, half on a fat-rich diet and half on a carb-rich diet, within just 2 days, this is what happens. The glucose intolerance skyrockets in the fat group. In response to the same sugar challenge, the group that had been shoveling in fat ended up with twice the blood sugar. As the amount of fat in the diet goes up, so does our blood sugar spikes. Why would eating fat lead to higher blood sugar levels? It would take scientists nearly 7 decades to unravel this mystery, but it would end up holding the key to our current understanding of the cause of type 2 diabetes. The reason athletes carb load before a race is to build up the fuel supply within their muscles. We break down the starch into glucose in our digestive tract. It circulates as blood glucose, also known as blood sugar, and is taken up by our muscles to be stored and burned for energy. Blood sugar, though, is like a vampire. It needs an invitation to come into our cells, and that invitation is insulin. Here's a muscle cell. Here's some blood sugar outside, waiting patiently to come in. Insulin is the key that unlocks the door to let sugar in our blood enter the muscle cell. When insulin attaches to the insulin receptor, it activates an enzyme, which activates another enzyme, which activates 2 more enzymes, which finally activates glucose transport, which acts as a gateway for glucose to enter the cell. So insulin is the key that unlocks the door into our muscle cells. What if there was no insulin, though? Well, blood sugar would be stuck out in the bloodstream, banging on the door to our muscles, and not be able to get inside, and so with nowhere to go, sugar levels would rise and rise. That's what happens in type 1 diabetes. The cells in the pancreas that make insulin get destroyed, and without insulin, sugar in the blood can't get out of the blood into the muscles, and blood sugar rises. But there's a second way we could end up with high blood sugar. What if there's enough insulin, but the insulin doesn't work? The key is there, but something's gummed up the lock. This is called insulin resistance. Our muscle cells become resistant to the effect of insulin. What's gumming up the door locks in our muscle cells, preventing insulin from letting sugar in? Fat. What's called intramyocellular lipid, fat inside our muscle cells. Fat in the bloodstream can build up inside the muscle cell, creating toxic fatty breakdown products and free radicals that can block the signaling pathway process. So no matter how much insulin we have out in our blood, it's not able to open the glucose gates, and blood sugar levels build up in the blood. This mechanism by which fat induces insulin resistance wasn't known until fancy MRI techniques were developed to see what was happening inside people's muscles as fat was infused into their bloodstream. That's how we found out that elevation of fat levels in the blood causes insulin resistance by the inhibition of glucose transport into the muscles. And this can happen within 3 hours. One hit of fat can start causing insulin resistance, inhibiting glucose uptake after just 160 minutes. Same thing happens to teens. You infuse fat into their bloodstream, it builds up in their muscles and decreases their insulin sensitivity, showing that increased fat in the blood is an important contributor of insulin resistance. And then you can do the opposite experiment. Lower the level of fat in people's blood, and the insulin resistance comes right down. Clear the fat out of the blood, and you can clear the sugar out of the blood. So that explains this finding. On a high-fat, ketogenic diet, insulin doesn't work very well. Our bodies become insulin resistant. But as the amount of fat in our diet gets lower and lower, insulin works better and better. This is a clear demonstration that the sugar tolerance of even healthy individuals can be impaired by administering a low-carb, high-fat diet. But we can decrease insulin resistance by decreasing fat intake. Free fatty acids, meaning free fat circulating in the bloodstream, not packaged into triglycerides, results in inflammation, toxic fat breakdown products, and oxidative stress, which can gum up the insulin receptor pathway and lead to insulin resistance in our muscles. And insulin resistance is what causes prediabetes and type 2 diabetes. As the level of fat in the blood rises, the body's ability to clear sugar from our blood drops. Where is this fat in our blood that's wreaking all this havoc coming from? It comes from the fat that we eat and the fat that we wear. The number of fat cells we have stays constant in adulthood. It's interesting. The way they figured this out is by measuring the amount of radioactive carbon trapped in our DNA from all the nuclear bomb tests. Anyway, after massive weight loss, our fat cells shrink as they offload fat, but the number stays the same. Conversely, when we gain weight, our fat cells just stretch as we pack more and more fat into each individual fat cell. So when our belly button thighs gets big, we're not adding more fat cells, we're just cramming more fat into each cell. And at a certain point, our fat cells become so bloated that they spill fat back into the bloodstream. This is an illustration of the so-called spillover effect. Not only does an obese person have more fat on their bodies, but they're constantly spilling that fat into their bloodstream. So that could be the link between obesity and diabetes. Fat is spilling out from our fat cells and gets lodged in our muscle cells, leading to the insulin resistance that promotes the onset of type 2 diabetes. Or the fat can enter our bloodstream through our mouth. If you put people on a low-carb diet, fat builds up in their muscles within 2 hours, compared to a low-fat diet, and insulin sensitivity drops. And the more fat in the muscle, the lower the ability to clear sugar from the blood. It doesn't take years for this to happen. Just hours after fatty foods go into our mouths, our body has problems using insulin. A fat-rich diet can increase fat in the blood, and this increase is accompanied by a decrease in insulin sensitivity. Studies clearly demonstrate that the fat in the blood directly inhibits blood sugar transport and usage in our muscles, which is responsible for clearing about 85% of the sugar out of our blood. These findings also indicate an important role of nutrition, particularly increased consumption of fat, for the development of insulin resistance. Normally, we only have about 10 to 50 micromoles of free fat floating around in our bloodstream at any one time. But those who are obese are constantly spilling fat out into their bloodstream, but we can reach those same levels in our blood eating a high-fat diet. So a skinny person eating a low-carb diet can have the same level of fat in their blood as obese people do. Similarly, being obese is like eating some horrible bacon and butter diet all day, because obese persons are constantly spilling fat into their bloodstream no matter what goes in their mouth. The association between fat and insulin resistance is now widely accepted, so-called ectopic fat accumulation, the accumulation of fat in places it's not supposed to be, like within our muscle cells. But all fats don't affect muscles the same. The type of fat, saturated versus unsaturated, is critical. Saturated fats like palmitate, found mostly in meat, dairy, eggs, cause insulin resistance, but oligate, found mostly in nuts, olives, avocados, may actually improve insulin sensitivity. What makes saturated fat bad? Saturated fat causes more of those toxic breakdown products and mitochondrial dysfunction, increased oxidative stress, free radicals, inflammation, causing a vicious cycle of events in which saturated fat-induced free radical causes dysfunction in the little power plants within our muscle cells, which cause an increase in free radical production and impairment of insulin signaling. Fat cells filled with saturated fat activate an inflammatory response to a far greater extent. This increased inflammation, along with eating more saturated fat, has been demonstrated to cause insulin resistance through free radical and ceramide production. Saturated fat has also been shown to have a direct effect on skeletal muscle insulin resistance. Accumulation of saturated fat increases the amount of diacylglycerol in our muscles, which has been demonstrated to have a potent effect on muscle insulin resistance. And it doesn't matter if the muscle in our blood, and the fat in our blood comes from our own fat or from their fat. You can take muscle biopsies from people and correlate the saturated fat buildup in their muscles with insulin resistance. While monounsaturated fats are more likely to be detoxified or safely stored away, saturated fats create these toxic breakdown products like ceramide that cause lipotoxicity. Lipo meaning fat, as in liposuction, and toxicity. This fat toxicity in our muscles is a well-known concept in the explanation of the trigger for insulin resistance. I've talked about the role of saturated and trans fats contributing to the progression of other diseases, like autoimmune diseases, cancer, heart disease. They can also cause insulin resistance, the underlying cause of prediabetes and type 2 diabetes. In the human diet, saturated fats are derived from animal sources, while trans fats originate in meat and milk in addition to partially hydrogenated and refined vegetable oils. That's why experimentally shifting people from animal fats to plant fats can improve insulin sensitivity. Insulin sensitivity was impaired on the diet with added butterfat, but not on the diet with added olive fat. We know prolonged exposure of our muscles to high levels of fat leads to severe insulin resistance, with saturated fats demonstrated to be the worst. But they don't just lead to inhibition of insulin signaling, the activation of inflammatory pathways, and the increase in free radicals. They cause an alteration in gene expression, leading to a suppression of key mitochondrial enzymes like carnitine palmitoyltransferase, which finally solves the mystery of why those eating vegetarian have 60% higher expression of that fat-burning enzyme. They're eating less saturated fat. So, do those eating plant-based diets have less fat clogging their muscles and less insulin resistance too? Well, there hasn't been any data available regarding the insulin sensitivity or inside muscle cell fat of those eating vegan or vegetarian until now. Researchers at the Imperial College of London compared the insulin resistance and muscle fat of vegans versus omnivores. Now, those eating plant-based diets have the unfair advantage of being so much slimmer, so they found omnivores who were as skinny as vegans to see if plant-based diets had a direct effect, as opposed to indirectly pulling fat out of the muscles by helping people lose weight in general. They found significantly less fat trapped in the muscle cells of vegans compared to omnivores, even at the same body weight. They found better insulin sensitivity, better blood sugar levels, better insulin levels, and, excitingly, significantly improved beta cell function, the cells in the pancreas that make the insulin in the first place. They conclude that eating plant-based is not only expected to be cardioprotective, helping prevent our #1 killer, heart disease, but that it may be beta cell protective as well, right? Helping also to prevent our 7th leading cause of death, diabetes. The food, alcohol, and tobacco industries have been blamed for manufacturing epidemics of chronic disease, but they're just trying to sell more product like everyone else. And so if that means distorting science, creating funk groups, compromising scientists, blocking public health policies, they're just trying to protect their business. It's not about customer satisfaction, but shareholder satisfaction. How else could we have tobacco companies, for example, continuing to produce products that kill 1 in 2 of their most loyal customers? Civil society organizations concerned with public health have earned a reputation for being anti-industry, but the issue is not industry, but that sector of industry whose products are harmful to public health. We like the broccoli industry. In fact, the corporate world might end up leading the lifestyle medicine revolution. The annual cost attributable to obesity alone among full-time employees is estimated at $70 billion, primarily because obese employees are not as productive on the job. Having healthy employees is good for the bottom line. Every dollar spent on wellness programs may offer a $3 return on investment. And if you track the market performance of companies that strive to nurture a culture of health, they appear to outperform their competition. That's why companies like Geico are exploring workplace dietary interventions. The remarkable success at Geico headquarters led to an expansion of the program at corporate offices across the country, with test sites from San Diego to Macon, Georgia. Given that previous workplace studies have found that workers who ate a lot of animal protein had nearly 5 times the odds of obesity, whereas those that ate the most plant protein appear protected, obese and diabetic employees were asked to follow a plant-based diet of whole grains, vegetables, beans, split peas, chickpeas, and lentils, and fruit, while avoiding meat, dairy, and eggs. Compliance wasn't great. Fewer than half really got their animal product consumption down, but there were definite improvements, significant reductions in saturated fat, an increase in protective nutrients. But enough to make a difference? Yes. Weight dropped, blood cholesterol dropped, and better blood sugar control in diabetics. And this was with no calorie counting, no portion control, no exercise component. The weight reduction appears to result from feeling fuller earlier due to higher dietary fiber intake. The difference in weight loss could also be the result of an increase in the thermic effect of food, allowing a small extra edge for weight loss in the vegan group. Those eating plant-based diets tend to burn off more calories in heat. Eating plants appears to boost metabolism. This may be due to increased insulin sensitivity in our cells, allowing cells to metabolize carbohydrate more quickly rather than storing it as body fat, and as a result, vegan diets have been shown to increase postprandial calorie burn by about 16%, up to 3 hours after consuming a meal. One sign of changing U.S. demographics is that salsa has replaced ketchup as America's #1 table condiment, one of the most popular ingredients of which is cilantro, one of the most polarizing and divisive food ingredients known. Some people love it, some people hate it. What's interesting is that the lovers and the haters appear to experience the taste differently. Individuals who like cilantro may describe it as fresh, fragrant, or citrusy, whereas those who dislike cilantro report that it tastes like soap, mold, dirt, or bugs. I don't know how people know what bugs taste like, but rarely are polarizing opinions about flavors so extreme. Maybe it's genetic. Different ethnic groups do seem to have different rates of cilantro dislike, with Ashkenazi Jews scoring highest on the cilantro hate-o-meter. Another clue came from twin studies that show that identical twins tend to share cilantro preferences, whereas regular fraternal twins do not have such a strong correlation. Our genetic code is so big, though, containing about 3 billion letters, that to find some cilantro gene, you'd have to analyze the DNA and cilantro tastes of like 10,000 people, and obviously genetic researchers have better things to do. Or maybe not. Here we present the results of a genome-wide association study among 14,000 participants who report whether cilantro tasted soapy, with replication in a distinct set of 11,000 people who declared whether they liked cilantro or not. And lo and behold, they found a spot on chromosome 11 that seemed to match. What's there? A gene called OR6A2 that enables us to smell certain chemicals like E2-decanol, a primary constituent of cilantro, and also the defensive secretions of stink bugs. So maybe cilantro does taste like bugs. But cilantro lovers may be genetic mutants that have an inability to smell the unpleasant compound. But that may actually be an advantage, though, since cilantro is healthy stuff. In fact, that's the justification to do these kinds of studies, to see why some people don't like the taste of healthy foods. Are the cilantro haters really missing out on much, though? Mother Nature has been described as the oldest and most comprehensive pharmacy of all time, and cilantro, called coriander around most of the world, is one of nature's oldest herbal prescriptions, credited with antimicrobial, antioxidant, anti-diabetic, anti-anxiety, anti-epilepsy, etc. properties. But these are all from preclinical studies, meaning on cells in a test tube or lab rats, studies like the anti-despair activity of cilantro in mice, testing animals in a despair apparatus. Finally, though, a human study on the anti-arthritis potential of cilantro. There was a study performed in Germany of a lotion made out of cilantro seeds, showing it could decrease the redness of a sunburn. showing it had some anti-inflammatory effects, though not as much as an over-the-counter steroid cream, hydrocortisone, or a prescription-strength steroid cream. Well, if the cilantro plant is anti-inflammatory, let's give it to some people with osteoarthritis and see if it helps. They gave about 20 sprigs of cilantro daily for 2 months and reported a significant drop in ESR in the cilantro group, a nonspecific indicator of inflammation. How did the patients do clinically, though? They didn't say, but did report a rather remarkable decrease in uric acid levels, a 50% drop, suggesting that huge amounts of cilantro may be useful for those suffering from a different arthritis called gout. Each of us has about 6 billion miles of DNA. How does our body keep it from getting all tangled up? There are special proteins called histones that act like spools with DNA as the thread. Enzymes called sirtuins wrap the DNA around the histones, and by doing so silence whatever genes were in that stretch of DNA, hence their name, sirtuins, which stands for Silencing Information Regulator. Although they were discovered only about a decade ago, the study of sirtuins has become one of the most promising areas of biomedicine. They appear to be involved in promoting healthy aging and longevity. Suppression of this key host defense is considered a central feature of Alzheimer's disease. Autopsies of Alzheimer's victims reveal that loss of sirtuin activity is closely associated with the accumulation of plaques and tangles in the brains that's characteristic of Alzheimer's disease. Sirtuins appear to activate pathways that steer the brain away from the formation of plaque and tangle proteins. Because a decrease in sirtuin activity can clearly have deleterious effects on nerve health, they're trying to come up with brains to increase sirtuin activity. But why not just prevent its suppression in the first place? Glycotoxins in our food suppress sirtuin activity. These so-called advanced glycation end products, or AGEs. Our modern diet includes excessive AGEs, which can be neurotoxic. High levels in the blood may predict cognitive decline over time. If you measure the urine levels of glycotoxins flowing through the bodies of older adults, those with the highest levels went on to suffer the greatest cognitive decline of the subsequent 9 years. As we age, our brain literally shrinks. In our 60s and 70s, we lose an average of 5 cubic centimeters of total brain tissue volume every year. But some lose more than others. Brain atrophy may be reduced in very healthy individuals, and in a few people don't lose any brain at all. Normally we lose about 2% of brain volume every year, but that's just the average. Although the average brain loss for folks in their 70s and 80s is 2.1%, some lose more, some lose less, and some men and women lose none at all over the period of 4 years. Researchers in Australia provided the first evidence linking AGEs with this kind of cerebral brain loss. So, limiting one's consumption of these compounds may end up having significant public health benefits. Because sirtuin deficiency is both preventable and reversible by dietary AGE reduction, a therapeutic strategy that includes eating less AGEs may offer a new strategy to combat the epidemic of Alzheimer's. Now, some glycotoxins are produced internally, particularly in diabetics, but anyone can get them from smoking and eating, particularly foods high in fat and protein. In a previous video, I listed the 15 foods most contaminated with glycotoxins. It turns out mostly chicken, but also pork, beef, and fish, which may help explain why those that eat the most meat may have triple the risk of getting dementia compared to longtime vegetarians. No matter where we live, how old we are, what we look like, health researchers have discovered that 90% of having a first heart attack can be attributed to 9 modifiable risk factors. The 9 factors that are threatening our lives include smoking, too much bad cholesterol, high blood pressure, diabetes, abdominal obesity, stress, a lack of daily fruit and vegetable consumption, as well as a lack of daily exercise. But Dr. William Clifford Roberts, executive director of the Baylor Heart and Vascular Institute and longtime editor-in-chief of the American Journal of Cardiology, is convinced that atherosclerosis has a single cause, namely the cholesterol, and that the other so-called atherosclerotic risk factors are only contributory at most. In other words, we could be stressed, overweight, smoking, diabetic couch potatoes, but if our cholesterol is low enough, there may be just not enough cholesterol in our bloodstream to infiltrate our artery walls and trigger the disease. Thus, the only absolute prerequisite for a fatal or non-fatal atherosclerotic event like a heart attack is an elevated cholesterol level. It was not appreciated until recently that the average blood cholesterol level in the United States, the so-called normal level, was actually abnormal, accelerating the blockages in our arteries and putting a large fraction of the normal population at risk. It's our #1 killer. That's cited as one of the reasons the cholesterol controversy lasted for so long, an unwillingness to accept the notion that a very large fraction of our population actually has an unhealthily high cholesterol level. Normal cholesterol levels may be terminal cholesterol levels. The optimal cholesterol level, the optimal bad cholesterol LDL level, is 50 to 70. Accumulating data from multiple lines of evidence consistently demonstrate that that's where a physiologically normal LDL level would be. That appears to be the threshold above which atherosclerosis and heart attacks develop. That's what we start out at birth with, that's what our fellow primates have, that's the level seen in populations free of the heart disease epidemic. But we can also look at all the big randomized controlled cholesterol-lowering trials. This is graphing the progression of atherosclerosis versus LDL cholesterol. More cholesterol, more atherosclerosis. But if you draw a line down through the points, you can estimate that the LDL level at which there is zero progression is down around an LDL cholesterol of 70. You can do the same with studies preventing heart attacks in the first place. Zero coronary heart disease events might be reached down around 55, and those who've already had a heart attack and trying to prevent a second one might need to push their LDL levels even lower. Atherosclerosis is endemic in our population, in part because the average person's LDL cholesterol is up around 130, approximately twice the normal physiologic level. The reason the federal government doesn't recommend everyone shoot for at least even just under 100 is that despite the lower risk accompanying more optimal cholesterol levels, the intensity of clinical intervention required to achieve such levels for everyone in the population would financially overload the healthcare system. Drug usage would rise enormously. But they're assuming drugs are the only way to get our LDL that low. But those eating really plant-based diets may hit the optimal cholesterol target without even trying, just naturally nailing under 70. The reason given by the federal government for not advocating what the science shows is best was that it might frustrate the public, who would have difficulty maintaining a lower level. But maybe the public's greatest frustration would come from not being informed of the optimal diet for health. What has driven the dramatic increase in prevalence of the inflammatory bowel disease, Crohn's disease, in societies that are rapidly westernized, a disease practically unknown just a century ago? What has truly changed in our internal and external environment that has led to the appearance of this horrible disease? Well, Japan suffered one of the most dramatic increases, and of all of the changing dietary components, animal protein appeared to be the strongest factor, an exponential increase in newly diagnosed Crohn's patients and daily animal protein intake, whereas the more vegetable protein, the fewer the cases of Crohn's, which is consistent with data showing a more plant-based diet may be successful in both preventing Crohn's disease and in treating Crohn's disease. But what about inflammatory bowel diseases in general? The largest study of its kind, 60,000 people followed for over a decade, found that high total protein intake, specifically animal protein, was associated with a significantly increased risk of the other big inflammatory bowel disease as well, called ulcerative colitis. Not just protein in general, the association between high protein intake and inflammatory bowel disease risk was restricted to animal protein intake. And since World War II, animal protein intake has increased not only in Japan, but also in all developed countries. This increase in animal protein consumption may explain some of the increased incidence of inflammatory bowel disease in the second half of the 20th century. And it's not just this study, that's about what most other studies found as well. But why? What's the difference between animal protein and plant protein? Well, animal proteins tend to have more sulfur-containing amino acids, like methionine, which bacteria in our gut can turn into the toxic rotten egg smell gas, hydrogen sulfide. Emerging evidence suggests that sulfur compounds may play a role in the development of ulcerative colitis, this chronic inflammatory bowel disease of the colon and rectum characterized by bloody diarrhea. The first hint as to the importance of our gut flora in general was in the 1970s, when analysis of stools showed that their bulk was made up of mostly bacteria, not undigested material. Most of our stool by weight is pure bacteria. We're pushing out trillions of bacteria a day. They just keep multiplying and multiplying, and they do wonderful things for us, like create the protective compound butyrate from the fiber we eat. But it depends on what we feed them. Bacteria may also elaborate toxic products from food residues such as hydrogen sulfide in response to a high-meat diet. Hydrogen sulfide is a bacterially derived cell poison that has been implicated in ulcerative colitis. We'd always assumed that sulfide generation in the colon is driven by dietary components such as sulfur-containing amino acids, but we didn't know for sure until this study. They had folks eat 5 different diets, each with escalating meat content from vegetarian all the way up to like a steak a day. And the more meat, the more sulfide. 10 times more meat meant 10 times more sulfide. They conclude that dietary protein from meat is an important substrate for sulfide generation by bacteria in the human large intestine. Hydrogen sulfide can then act as a free radical and damage our DNA at concentrations way below what our poor colon lining is exposed to on a routine basis, which may help explain why diets higher in meat and lower in fiber may produce so-called fecal water that causes about twice as much DNA damage. Fecal water is like when researchers make a tea from someone's stool. The biology of sulfur in the human gut has escaped serious attention until recently. Previously, it was just thought of as the rotten egg smell and malodorous intestinal gas. But the increase in sulfur compounds in response to a supplement of animal protein is not only of interest in the field of flatology, which is the formal study of farts, but may also be of importance in the development of ulcerative colitis. More than 30 years ago, an idea was put forward that high colonic pH promoted colorectal cancer. High colonic pH may promote the creation of carcinogens from bile acids, a process that's inhibited once you get below a pH of about 6.5. This is supported by data like this, showing those at higher risk for colon cancer may have a higher stool pH, and those at lower risk a lower pH. Dramatic difference between the two groups, with most of the high-risk group a pH over 8, and most of the low-risk group pH under 6. This may explain the 50-fold lower rates of colon cancer in Africa compared to America. The bacteria we have in our gut depends on what we eat. If we eat a lot of fiber, then we preferentially feed the fiber-eating bacteria, which give us back all sorts of health-promoting substances like short-chain fatty acids that have anti-inflammatory and anti-cancer properties. More of these organic acids in the stools of native Africans than in African Americans. More acids, so lower pH, whereas putrefactive bacteria eating animal protein are able to increase stool pH by producing alkaline metabolites like ammonia. The pH of the stools of white versus black children in Africa was compared. Children, because you can more readily sample their stools, particularly the rural black school children who were eating such high-fiber diets, whole grains, legumes, nuts, vegetables, fruits, and wild greens, 9 out of 10 could produce a stool on demand. Stuffed from head to tail with plants, they could give you a stool sample at any time, as easy as getting a urine sample. Hard to even get access to the white kids, though, who were reluctant to participate in such investigations, even though they were given waxed cartons fitted with lids, and all the black kids got was a plate and a square of paper towel. What'd they find? Significantly lower fecal pH in those eating the traditional rural plant-based diets compared to those eating the Western diet, who were eating far fewer whole plant foods than the black children. But remove some of those whole plant foods, like switch their corn for white bread for just a few days, and their stool pH goes up, and add more whole plant foods, like an extra 5 to 7 servings of fruit every day, and their stool pH goes down even further, gets more acidic. It makes sense, right? I mean, what happens when you ferment plants, fruits, veggies, grains? They turn sour, like vinegar, sauerkraut, sourdough, right? Because good bacteria like lactobacillus produce organic acids like lactic acid, and those who eat lots of plants have more of these good bugs in their system. So using the purple cabbage test, we want blue pea, but pink poo. No surprise, then, if you compare the fecal samples of those eating vegetarian or vegan to those eating standard diets, plant-based diets appear to shift the makeup of the bacteria in our gut, resulting in a significantly lower stool pH. And the more plant-based, the lower the pH dropped. It's like a positive feedback loop. Fiber-eating bacteria produce the acids to create the pH at which fiber-eating bacteria thrive, while suppressing the group of less beneficial bugs. How long does it take to bring stool pH down on a plant-based diet? As little as 2 weeks. A dozen volunteers carefully selected for their trustworthiness and randomized to sequentially go on regular, vegetarian, or vegan diets, and 2 weeks in, a significant drop in fecal pH was achieved eating completely plant-based. But there are plant-based diets, and then there are plant-based diets. Remember, these two groups, dramatically different stool pH, yet both groups were vegetarian. But the high-risk group was eating mostly refined grains, very little fiber, whereas the low-risk group was eating whole grains and beans packed with fiber for a fiber-friendly flora to munch on. Just as a reduction in high serum cholesterol contributes to the avoidance of coronary heart disease, so a fall in the fecal pH value may contribute to the avoidance of bowel cancer, and through the same means, right? Eating more whole plant foods. The Mediterranean diet is an in topic nowadays in both the medical literature and the lay media. 450 papers published in the medical literature over just the last year alone, more than one a day. Uncritical laudatory coverage is common, but specifics are hard to come by. What is it? Where did it come from? Why is it good? Merits are rarely detailed, possible downsides never mentioned, so let's dig in. After World War II, the government of Greece asked the Rockefeller Foundation to come in and assess the situation. Impressed by the low rates of heart disease in the region, nutrition scientist Ancel Keys, after which K-rations were named, initiated his famous 7 countries study, in which he found the rate of fatal heart disease on the Greek Isle of Crete was 20 times lower than that in the United States. They also had the lowest cancer rates and fewest deaths overall. What were they eating? Their diets were more than 90% plant-based, which may explain why coronary heart disease was such a rarity. A rarity, that is, except for a small class of rich people whose diet differed from that of the general population. They ate meat every day instead of every week or two. So the heart of the Mediterranean diet is mainly vegetarian, much lower in meat and dairy, which Keys considered the major villains in the diet because of their saturated fat content. Unfortunately, no one is really eating the traditional Mediterranean diet anymore, even in the Mediterranean. The prevalence of coronary heart disease skyrocketed by an order of magnitude within a few decades in Crete, blamed on the increased consumption of meat and cheese at the expense of plant foods. Everyone is talking about the Mediterranean diet, but few are those who do it properly. People think pizza or spaghetti with meat sauce, but while Italian restaurants brag about the healthy Mediterranean diet, they serve a travesty of it. So if no one's really eating this way anymore, how do you study it? researchers came up with a variety of Mediterranean diet adherence scoring systems to see if people who are eating more Mediterranean-ish do better. You get maximum points the more plant foods you eat, but effectively get points deducted eating just a single serving of meat or dairy a day. So no surprise, those that eat relatively higher on the scale have a lower risk of heart disease, lower risk of cancer, and lower risk of death overall. After all, the Mediterranean diet can be considered to be a near-vegetarian diet. As such, it should be expected to produce the well-established health benefits of vegetarian diet. So less heart disease, cancer, and death, and less inflammation, an improved arterial function, a lower risk of developing type 2 diabetes, and a reduced risk for stroke, depression, and cognitive impairment. How might it work? I've talked about the elegant studies showing that those who eat plant-based diets have more plant-based compounds, like aspirin, circulating within their systems. Polyphenol phytonutrients in plant foods are associated with significant lower risk of dying in and of themselves. Magnesium consumption also associated with significant lower risk of dying. The magnesium found in dark green leafy vegetables, as well as fruits, beans, nuts, soy, and whole grains. Heme iron, on the other hand, the iron found in blood and muscle, acts as a pro-oxidant and appears to increase the risk of diabetes, whereas plant-based non-heme iron appeared safe. Same thing with heart disease. Animal-based iron found to significantly increase the risk of coronary heart disease, our #1 killer, but not plant-based iron. The Mediterranean diet is protective compared to the standard American diet, no question, but any diet rich in whole plant foods and low in animal fat consumption could be expected to confer protection against many of our leading killers. Recent studies have shown that higher Mediterranean diet adherence scores are associated with a significant reduction in risk of death, heart disease, cancer, and brain disease. But the problem with population studies like these is that people who eat healthier may also live healthier, and so how do we know it's their diet? As the American Heart Association position states, before advising people to follow a Mediterranean diet, we need more studies to find out whether the diet itself or other lifestyle factors account for the lower deaths from heart disease. How do you do that? Well, there are ways you can control for obvious things like smoking and exercise, which many studies did, but ideally you do an interventional trial, the gold standard of nutritional science. Take people, change their diet, while trying to keep everything else the same, and see what happens. And that's what we got 20 years ago, the famous Lyon Diet Heart Study. About 600 folks who had just had their first heart attack were randomized into two groups. The control group got no dietary advice, apart from whatever their doctors were telling them. But the experimental group was told to eat more of a Mediterranean-type diet, supplemented with a canola oil-based spread to give them the plant-based omega-3s they normally beget from eating weeds and walnuts if they actually lived on a Greek isle in the 1950s. The Mediterranean diet group did end up taking some of the dietary advice to heart. They ate more bread, more fruit, less deli meat, less meat in general, and less butter and cream. But other than that, no significant changes in diet reported in terms of wine, olive oil, or fish consumption. So, less saturated fat and cholesterol, more plant-based omega-3s, but not huge dietary changes. But at the end of about 4 years, in the control group, 44 individuals had a second heart attack, either fatal or non-fatal, but only 14 suffered another attack in the group that changed their diet. So they went from having like a 4% chance of having a heart attack every year down to like 1%. Now, a cynic might say, yes, less death and disease, but the Mediterranean diet continued to feed their heart disease, so much so that 14 of them suffered new heart attacks while on the diet. Now, their disease progressed a lot less than the regular diet group, about 4 times less, but what if there was a diet that could stop or reverse heart disease? Dr. Caldwell Esselstyn and colleagues at the Cleveland Clinic recently published a case series of 198 consecutive patients with cardiovascular disease counseled to switch to a diet composed entirely of whole plant foods. Of the 198, 177 stuck to the diet, whereas the other 21 fell off the wagon, setting up kind of a natural experiment. What happened to the 21? This was such a sick group of patients that more than half suffered from a fatal heart attack or needed angioplasty or a heart transplant. In that same time period of about 4 years, of the 177 that stuck to the plant-based diet, only one had a major event as a result of worsening disease. Not half, but less than 1%. As Dean Ornish noted in his response to the latest Mediterranean trial, a Mediterranean diet is better than what most people are consuming, but even better may be a diet based on whole plant foods. Now, this was not a randomized trial, so can't directly be compared to the Lyon study, and included very determined patients. Not everyone is willing to dramatically change their diets, even if it may literally be a matter of life or death, in which case, rather than doing nothing, eating a more Mediterranean-type diet may cut risk for heart attack survivors by about 2/3. Cutting 99% of risk would be better if Esselstyn's results were replicated in a controlled trial, but even a 70% drop in risk could save tens of thousands of lives every year. The Lyon Diet Heart trial showed that a Mediterranean-type diet could significantly reduce the risk of having a second heart attack, but since many first heart attacks are fatal, better to prevent heart attacks in the first place. But no randomized trial had ever been conducted to test the Mediterranean diet for this so-called primary prevention until now. The PREDIMED study from the Spanish Prevención con Dieta Mediterránea, in which a whopping 7,447 patients were randomized into 3 groups. These were folks at high risk for a heart attack, about half were obese, diabetic, most had high blood pressure, high cholesterol, but had not yet had their first heart attack or stroke. A third were told to eat a Mediterranean diet and given a free quart of extra virgin olive oil every week. The second group was told to eat a Mediterranean diet and given a half pound of free nuts every week. And the third group was told to follow the American Heart Association guidelines and reduce their fat intake. No portion control or exercise advice was given, and they were followed for about 5 years. The results were published in the prestigious New England Journal of Medicine. Now, the first thing you do when you look at a diet intervention trial is you see what the groups actually ended up eating, which is often very different from what they were told to eat. For example, the so-called low-fat group started out at 39% of calories from fat and ended up with 37% of calories from fat, which is high fat, even compared to the standard American diet, which comes in at 33%. Something the researchers plainly acknowledged. In fact, the control group didn't change their diets much at all over the years, so can be thought of as the what if you don't do anything group, which is still an important control group to have, though the 2 Mediterranean diet groups didn't get much more Mediterranean. They were told to eat more fruits and vegetables, for example, less meat and dairy, but didn't accomplish either of those goals compared to control. The biggest changes reported were, not surprisingly, in the consumption of the freebies. The group that got a free jug of extra virgin olive oil delivered to their home every week really did start increasing their consumption, in part by replacing some of the refined olive oil they had been using, but of course would have to pay for. And those that got a half pound of free nuts sent to them every week for 4 years straight did start eating more nuts. Too bad they couldn't have slipped a little free broccoli in there too, but so basically they designed a study to test 2 different Mediterranean diets versus a low-fat diet, but ended up studying something very different. In essence, what happens when thousands of people switch from consuming about 3 tablespoons of olive oil a day, half virgin, to 4 tablespoons of all virgin, compared to thousands of people who all of a sudden go from eating about a half an ounce of nuts a day to a whole ounce, compared to thousands of people who don't make much of a change at all. It may not have been what they were hoping for, but these are important research questions in and of themselves. Let's say you're at high risk for heart disease, eating like this. What would happen if you started to add an extra half ounce of nuts to your daily diet, or more unrefined olive oil? We didn't know until now. With no significant differences in meat and dairy intake, there were no significant differences in saturated fat or cholesterol intake, so no surprise, no significant differences in their blood cholesterol levels, and so No difference significantly in their subsequent number of heart attacks. In the 5 or so years the study ran, there were 37 heart attacks in the olive oil group, 31 in the nut group, 38 in the neither group, no significant difference. Same with dying from a heart attack or stroke, or dying from any cause. But those in the olive oil, and especially the nut group, had significantly fewer strokes. All 3 groups were eating stroke-promoting diets. They all had strokes in them after eating these diets for years, and so ideally, we choose diets that can stop or reverse the disease process. But the diet with the added extra virgin olive oil caused about a third fewer strokes, and adding nuts seemed to cut their stroke risk nearly in half. If this worked as well in the general population, in the U.S. alone, that would mean preventing 89,000 strokes a year. That would be like 10 strokes an hour around the clock prevented by simply adding half an ounce of nuts to one's daily diet. How might adding a few nuts to one's daily diet, in this case about 4 walnuts, almonds, and hazelnuts, cut one's stroke risk nearly in half? Well, olives and nuts are plant foods, and as such packed with antioxidants, raising the antioxidant level of our bloodstream, resulting in lower fat oxidation and free radical DNA damage. But what's happening inside people's arteries? Researchers measured the amount of atherosclerotic plaque in the neck arteries going to the brain in folks who for years were eating the added nuts or added extra virgin olive oil, or neither, to their daily diets. In the control group, the plaque got worse, which is what happens when one continues to eat an artery-clogging diet. But there were no significant changes in the added extra virgin olive oil group, and the plaque in the added nut group appeared to get better. The nuts appeared to induce a regression of the disease, or at least a significant delay in the progression. Now, the nut group was still suffering strokes, but only half as many, perhaps because the reduction in plaque height within the arteries on extra nuts was indicating a stabilization of the plaque, rendering them less likely to rupture. Adding nuts to our diet may also improve endothelial function, boosting the ability of our arteries to dilate naturally by about 30%. If you look at the baseline adherence to Mediterranean diet principles and control for things like smoking and exercise, there are only 2 factors significantly associated with reduced heart attack and stroke risk: more vegetables and more nuts. No significant association with the olive oil, or the wine, or the fish, or cutting back on soda and cookies. Among the individual Mediterranean diet components, only increasing consumption of vegetables and nuts were related to reduced cardiovascular events. On the one hand, cutting stroke risk in half just by eating a handful of nuts a day is pretty amazing, but those in the added nut group didn't appear to live any longer overall. And other studies had suggested that frequent nut consumption may indeed extend life. For example, the Harvard Health Professionals studies, involving a whopping 3 million person-years of follow-up over decades, found nut consumption associated with fewer deaths from cancer, heart disease, respiratory disease, and most importantly, fewer deaths overall, confirmed by all the other big major prospective studies. As of 2014, with a new one just published, 17,000 people followed for 5 years, and those who ate nuts had less than half the risk of dying. Maybe this is just because people who eat nuts instead of meat, eggs, and dairy, and that's why they live longer? Well, no, since even vegetarians who frequently eat nuts also have a dramatically reduced risk compared to those that don't. So what's going on here with the Predimed study showing no longevity benefit from nuts? Did they just not wait long enough? Well, just because people were randomized to the nut group doesn't mean they actually ate more nuts, and those randomized to the other groups didn't necessarily stay away from nuts. If you reanalyze the data comparing the death rates of just those who actually ate more nuts to those who actually didn't, nut consumption was indeed associated with a significantly reduced risk of death. If you do the same kind of post-hoc analysis with olive oil, even with the extra virgin, no benefit in terms of living longer. This is consistent with how Ancel Keys, the so-called father of the Mediterranean diet, viewed olive oil. He thought of its benefit more as a way of just replacing the animal fats, anything to get people to eat less lard and butter. What are the best kinds of nuts? Well, the greatest benefits were attributed to walnuts. Particularly, it seems, for preventing cancer deaths. Those eating more than 3 servings of walnuts a week appear to cut the risk of dying from cancer in half. Now it's just a matter of communicating the research to the public. All the major cancer groups emphasize a more plant-based diet, remarkably consistent with the World Health Organization guidelines for healthy eating. The far-reaching positive effects of a plant-based diet, including walnuts, may be the most critical message for the public. What accounts for the benefits of a Mediterranean-style diet? An Anatomy of Health Effects was published, and the single most important component was the high consumption of plant foods. In contrast, fish and seafood consumption, the only animal foods promoted in the Mediterranean diet, did not seem to help. In fact, if you look at the 4 major dietary quality scoring systems, which have all been associated with extending lifespan and lowering heart disease and cancer mortality, they all share only 4 things in common: more fruit, more vegetables, more whole grains, and more nuts and beans. They're all built on a common core of a diet rich in plant foods, whereas opposite food patterns, rich in animal foods, poor in plant-based foods, in other words, the Western diet, is associated with higher risks. So we need to optimize the food environment to support whole grains, vegetables, fruit, and plant-based proteins. That's one of the things all the so-called blue zones have in common, the longest living populations. Not only social support and engagement, daily exercise, but nutritionally, they all center their diet around plant foods, reserving meat mostly for special occasions. And the population with perhaps the longest life expectancy in the world doesn't eat any meat at all, the California Adventist vegetarians. So if the primary benefits of the Mediterranean diet are due to all the whole plant foods, what if you went back to the famous Predimed study and created a pro-vegetarian scoring system? We know vegetarians live longer, but because a pure vegetarian diet might not easily be embraced by many individuals, maybe it would be easier to swallow if we just tell people, look, more plant-based foods, less animal-based foods. But would just moving along the spectrum towards more plants actually enable people to live longer? They thought of this food pattern as a gentle approach to vegetarianism, figuring that if it improved survival, it would be an easily understandable message for health promotion. More plant foods, less animal foods. So you get points for eating fruits, vegetables, nuts, grains, beans, olive oil, and potatoes, but get docked points for any animal fats, eggs, fish, dairy, or any type of meat or meat products. Of course, that means you get a higher score the more potato chips and French fries you eat. That's why I prefer the term whole food plant-based diet, since it's defined by what you eat, not by what you don't eat. When I was teaching up at Cornell, I'd have vegan students apparently living off of French fries and beer. Vegan does not necessarily mean health-promoting. But did it work? Regardless of healthy versus unhealthy, if you give points to people for any kind of plant food, processed or not, and detract points for any kind of animal product consumption, do people with higher scores live longer? Yes. The maximum pro-vegetarian score is 60, but even just scoring 40 or more was associated with a 40% drop in mortality. In fact, there were so few deaths in the highest category of adherence to the pro-vegetarian diet, they had to merge the 2 upper categories for their analysis. This is evidence that simple advice to increase the consumption of plant-derived foods with reductions in the consumption of foods from animal sources confers a survival advantage, a live a longer life advantage. This modest change is realistic, affordable, and achievable because a sizable proportion of their population was already eating that way, so one can get a significant survival benefit without a radical shift to the exclusive consumption of plant foods, a more gradual and gentle approach more easily translatable into public policy. For example, a 41% drop in mortality rates in the United States would mean saving the lives of hundreds of thousands of Americans every year. The traditional Mediterranean diet can be considered as a mainly, but not exclusively, plant-based diet, and certainly not a whole food plant-based diet, as olive oil and wine can be considered essentially fruit juices. Even if one is eating a vegetarian diet, an entirely plant-based version, there are a number of potentially problematic nutritional aspects that are rarely talked about. For example, lots of white bread, white pasta, not a lot of whole grains. Alcohol can be a problem. The caloric density with all that oil can be high, as well as the salt intake. Let's go over these one by one. In this anatomy of the health effects of the Mediterranean diet, while the single most important component was the high consumption of plant foods, in contrast, high cereal consumption, meaning high grain consumption, did not appear to help. This may be because most grains modern Mediterranean dieters eat are refined, like white bread, whereas the traditional Mediterranean diet was characterized by unprocessed cereals, in other words, whole grains. And while whole grains have been associated with a lower risk of diabetes, heart disease, and cancer, refined grains may increase the risk of diabetes, obesity, heart disease, and other chronic diseases. For example, in the Predimed study, those who ate the most white bread, but not whole grain bread, gained significant weight. Alcohol may also be a problem. As a plant-centered diet, no surprise adherence to a Mediterranean diet is associated with lower cancer risk, but not with lower breast cancer risk. With all the fruits, veggies, nuts, seeds, beans, and low saturated fat, you'd assume there'd be a lower breast cancer risk. But alcohol is a known breast cancer risk factor, even moderate amounts. So if you create a special adapted version of the Mediterranean diet score that excludes alcohol, the diet does appear to reduce breast cancer risk then. The wonderful grape phytonutrients in red wine can improve our arterial function, such that if you drink de-alcoholized red wine with the alcohol removed, you get a significant boost in endothelial function, the ability of our arteries to relax and dilate normally, increasing blood flow. But if you drink the same red wine with the alcohol added back in, it abolishes the beneficial effect. It counteracts the benefit of the grape phytonutrients, so better just to eat grapes. Similarly, there are components of olives, the antioxidant phytonutrients that may help endothelial function. But when consumed as oil, even extra virgin olive oil may impair arterial function. So even if white bread dipped in olive oil is the very symbol of the Mediterranean diet, we can modernize it by removing oils and refined grains. Another important, albeit frequently ignored, issue in the modern Mediterranean diet is sodium intake. Despite evidence linking salt intake to high blood pressure, heart disease, and strokes, dietary salt intakes in the U.S. is on the rise. Right now we get about 7 to 10 grams a day, mostly from processed foods. If we were to decrease that just by 3 grams, which is about 1,000 milligrams of sodium, half a teaspoon of table salt every year, we could save tens of thousands of people from having a heart attack, prevent tens of thousands of strokes, and tens of thousands of deaths. There's a common misconception that only certain people should reduce their salt intake, and that for the vast majority of the population, salt reduction is unnecessary. But in reality, the opposite is true. But there's much we can learn from the traditional Mediterranean diet. A defining characteristic of the Mediterranean diet is an abundance of plant foods. One thing that seems to have fallen by the wayside: no real Mediterranean meal is replete without lots of greens, a key part of not only a good Mediterranean diet, but of any good diet. We've known for decades that even a single bout of exercise can elevate our mood, but enough to be used as a treatment for major depression? Well, we know physical activity has been associated with decreased symptoms of depression. For example, if you look at a cross-section of 8,000 people across the country, those that exercise regularly were less likely to have a major depression diagnosis. That's just a snapshot in time, though. If you look at that study, the researcher openly admits this may be a case of reverse causation. Maybe exercise didn't cut down on depression, maybe depression cut down on exercise. The reason depression may be associated with low physical activity is that people feel too lousy to get out of bed. What we need is an interventional study, where you take people who already have depression, randomize them into an exercise intervention, and see if they get better. And that's what we got. Men and women over 50, major depression, were randomized to either do an aerobic exercise program for 4 months or take an antidepressant drug called Zoloft. This is where they started out before, with Hamilton depression scores up around 18. Anything over 7 is considered depressed. But within 4 months, the drug group came down to normal, which is exactly what the drugs are supposed to do. What about the exercise-only group? No drugs. Same powerful effect. They conclude that an exercise training program may be considered an alternative to antidepressants for treatment of depression in older persons, given that they've shown that a group program of aerobic exercise is a feasible and effective treatment for depression, at least for older people. Not so fast, though. A group program? They have the exercise group folks come in 3 times a week for a group class. Maybe the only reason the exercise group got better is because they were forced to get out of bed, interact with people. Maybe it was the social stimulation and had nothing to do with the actual exercise. Before you could definitively say that exercise can work just as good as drugs, what you'd like to see is the same study, but with like an additional group, the same 2 plus a home exercise group, where they were just told to exercise on their own at home, no extra social interaction. But nothing like that had ever been done, until it was. The largest exercise trial of patients with major depression conducted to date, and not just including older persons, but other adults as well, and 3 different treatment groups this time. A home exercise group in addition to the supervised group exercise, and the drug group as before. And they all worked about just as well in terms of forcing the depression into remission. So we can say with confidence that exercise is comparable to antidepressant medication in the treatment of patients with major depressive disorder. Putting all the best studies together, the evidence indicates that exercise at least has a moderate antidepressant effect, and at best, exercise has a large effect on reductions in depressive symptoms, and could be characterized as a very useful and powerful intervention. Unfortunately, while studies support the use of exercise as a treatment for depression, exercise is rarely prescribed as a treatment for this common and debilitating problem. We've learned that exercise compares favorably to antidepressant medications as a first-line treatment for mild to moderate depression. But how much is that really saying, and how effective are antidepressant drugs in the first place? A recent meta-analysis has sparked huge scientific and public controversy by stating that the placebo effect can explain the apparent clinical effectiveness of antidepressants. But wait a second, aren't there clinical trials providing compelling evidence for antidepressant effectiveness? In fact, thousands of them? If a meta-analysis just compiles all the best published research, how could it say they don't work much better than sugar pills? The key word is published. What if a drug company decided only to publish studies that showed a positive effect, but quietly shelved and concealed any studies showing the drug didn't work? If you didn't know any better, you'd look at the published medical literature and be like, wow, this drug is great. And what if all the drug companies did it? To find out if this was the case, researchers applied to the FDA under the Freedom of Information Act to get access to the published and unpublished studies submitted by pharmaceutical companies, and what they found was shocking. According to the published literature, the results of nearly all the trials on antidepressants were positive. They work. In contrast, FDA analysis of the trial data showed that only roughly half of the trials had positive results. In other words, about half the studies showed the drugs didn't work. Thus, when published and unpublished data are combined, they fail to show a clinically significant advantage for antidepressant medication over a sugar pill. Not publishing negative results undermines evidence-based medicine, and puts millions of patients at risk for using ineffective or unsafe drugs, and this was the case for these antidepressant drugs. These revelations hit in 2008. Prozac, Surzone, Paxil, and Effexor. They worked, but so did sugar pills, and the difference between the drug and the placebo was small. That was 2008. Where were we by 2014? Analyses of the published data and the unpublished data that were hidden by the drug companies reveals that most, if not all, of the benefits of antidepressants are due to the placebo effect. And what's even worse, Freedom of Information Act documents show the FDA knew about it, but made an explicit decision to keep this information from the public and from prescribing physicians. How could drug companies get away with this? The pharmaceutical industry is considered the most profitable and politically influential industry in the United States, and mental illness can be thought of as the drug industry's golden goose, incurable, common, long-term, involving multiple medications. What could be better than that? Antidepressant medications are prescribed to 8.7% of the U.S. population. It's a multi-billion-dollar market, just those class of drugs. To summarize, there is a strong therapeutic response to antidepressant medication. It's just that the response to placebo is almost as strong. Indeed, antidepressants offer substantial benefits to millions of people suffering from depression. To cast them as ineffective is inaccurate. Just because they may not work better than fake pills doesn't mean they don't work. It's like homeopathy. Just because it doesn't work better than the sugar pills that they are doesn't mean that homeopathy doesn't work, because the placebo effect is real and is powerful. In this psychopharmacology journal, a psychiatrist funded by the Prozac company defends the drugs. A key issue is disregarded by the naysaying critics. If the patient is benefiting from antidepressant treatment, does it matter whether this is being achieved via drug or placebo effects? Yes, it matters. Among the side effects of antidepressants, sexual dysfunction up to like 3/4 of people. Long-term weight gain, insomnia, nausea, diarrhea. About 1 in 5 show withdrawal symptoms when they try to quit. And perhaps most tragically, they may make people more likely to become depressed in the future. People are more likely to become depressed again after treatment by antidepressants than after treatment by other means, including placebo. So if doctors are willing to give patients placebo-equivalent treatments, maybe it'd be better for them to just lie to patients and give them actual sugar pills. Yes, that involves deception, but isn't that preferable than deception with a side of side effects? Maybe medical bodies need to accept that a spoonful of deception may be therapeutic. If different treatments are equally effective, then shouldn't the choice be based on risk and harm? And of all the available treatments, antidepressant drugs may be among the riskiest and most harmful. If they are to be used at all, it should be as a last resort when depression is extremely severe and all of the treatment alternatives have been tried and failed. Antidepressants may not work better than placebo for mild and moderate depression, but for very severe depression, the drugs do beat out sugar pills. But that's just a small fraction of the people that are taking these drugs. That means that the vast majority of depressed patients, as many as 9 out of 10, are being prescribed medications that have negligible benefits to them. Too many doctors just quickly decide upon a depression diagnosis without necessarily even listening to what the patient has to say, and they end up putting them on antidepressant drugs without considering alternatives. Fortunately, there are effective alternatives. Physical exercise, for example, can have lasting effects, and hey, if it turns out to also be the placebo effect, it's at least a placebo with an enviable list of side effects. Whereas side effects of antidepressant drugs include things like sexual dysfunction and insomnia, side effects of exercise include enhanced libido and better sleep, decreased body fat, improved muscle tone, and a longer life. Milk is touted to build strong bones, but a compilation of all the best studies found no association between milk consumption and hip fracture risk, so drinking milk as an adult may not help bones. But what about in adolescence? Harvard researchers decided to put it to the test. Studies have shown that greater milk consumption during childhood and adolescence contributes to peak bone mass, and is therefore expected to help avoid osteoporosis and bone fractures in later life. But that's not what they found. Milk consumption during teenage years was not associated with a lower risk of hip fracture, and if anything, milk consumption was associated with a borderline increase in fracture risk in men. It appears that the extra boost in total body bone mineral density you get from getting extra calcium is lost within a few years, even if you keep the calcium supplementation up. This suggests a partial explanation for the long-standing enigma that hip fracture rates are highest in populations with the greatest milk consumption. Maybe an explanation why they're not lower, but why would they be higher? This enigma irked a Swedish research team, puzzled because studies, again and again, had shown a tendency of higher risk of fracture with a higher intake of milk. Well, there is a rare birth defect called galactosemia, where babies are born without the enzymes needed to detoxify the galactose found in milk, so they end up with elevated levels of galactose in their blood, which can cause bone loss even as kids. So maybe, the Swedish researchers figured, even in normal people that can detoxify the stuff, it might not be good for the bones to be drinking it every day. And galactose doesn't just hurt the bones. That's what scientists use to cause premature aging in lab animals. They slip them a little galactose, and you can shorten their lifespan, cause oxidative stress, inflammation, brain degeneration, just with like the equivalent of 1 to 2 glasses of milk's worth of galactose a day. We're not rats, though, but given the high amount of galactose in milk, recommendations to increase milk intake for prevention of fractures could be a conceivable contradiction, so they decided to put it to the test, looking at milk intake and mortality, as well as fracture risk, to test their theory. 100,000 men and women followed for up to 20 years. What did they find? Milk-drinking women had higher rates of death, more heart disease, significantly more cancer for each glass of milk. 3 glasses a day was associated with nearly twice the risk of death, and they had significantly more bone and hip fractures too. Men in a separate study also had a higher rate of death with higher milk consumption, but at least they didn't have higher fracture rates. So a dose-dependent higher rate of both mortality and fracture in women, and a higher rate of mortality in men with milk intake. But the opposite for other dairy products like soured milk and yogurt, which would go along with the galactose theory, since bacteria can ferment away some of the lactose. To prove it, though, we need a randomized controlled trial to examine the effect of milk intake on mortality and fractures. As the accompanying editorial pointed out, we better figure this out soon, as milk consumption is on the rise around the world. Rice currently feeds almost half the human population, making it the single most important staple food in the world. But a meta-analysis of 7 cohort studies following 350,000 people for up to 20 years found that higher consumption of white rice was associated with a significant increased risk of type 2 diabetes, especially in Asian populations. They estimated each serving per day of white rice was associated with an 11% increase in risk of diabetes, which may help explain why the association was even stronger in Asia, where they eat much more rice. This could explain why China has almost the same diabetes rates as we do. They have at about 10%, we're at about 11%, despite 7 times less obesity in China. Japan has 8 times less obesity than we do, yet may have a higher incidence of newly diagnosed diabetes cases than we do, 9 per 1,000 compared to our 8. They're skinnier and still may have more diabetes, maybe because of all the white rice they eat. Just like eating whole fruit is associated with a lower risk of diabetes, whereas eating fruit processed into juice may not just be neutral, but actually increase diabetes risk. Eating whole grains, like whole wheat bread or brown rice, is associated with a lower risk of diabetes, whereas eating white rice, a processed grain, may not just be neutral, but actually increase diabetes risk. White rice consumption does not appear associated with an increased risk of heart attack or stroke, though, which is a relief after this earlier study in China suggested a connection with stroke. But do we want to eat a food that's just neutral regarding some of our leading causes of death, when we can eat something associated with a lower risk of diabetes, heart attack, stroke, and weight gain? But if you look at the Cornell-Oxford China project, rural plant-based diets centered around rice were associated with relatively low risk of the so-called diseases of affluence, which includes diabetes. Maybe Asians just genetically don't get the same blood sugar spike when they eat white rice? No, if anything, people of Chinese ethnicity get higher blood sugar spikes. The rise in these diseases of affluence in China over the last half-century has been blamed in part on the tripling of the consumption of animal-source foods. The upsurge in diabetes has been most dramatic, and it's mostly just happened over the last decade. That crazy 9.7% diabetes prevalence figure that rivals ours is new. They appeared to have one of the lowest diabetes rates in the world in the year 2000. So what happened to their diets in the last 20 years or so? Well, oil consumption went up about 20%, pork consumption alone went up 40%, and rice consumption dropped about 30%. So diabetes rates were skyrocketing, while rice consumption was going down. So maybe it's the animal products and junk food that are the problem. Yes, brown rice is better than white rice, but to stop the mounting Asian epidemic, maybe we should focus on removing the cause, the toxic Western diet. That would be consistent with data showing animal protein and fat consumption associated with increased diabetes risk. But that doesn't explain this. If the rise in meat consumption is to blame, then why do the biggest recent studies in Japan and China associate white rice intake with diabetes? The answer to this puzzle may be that animal protein is making the rice worse. If you feed people mashed white potatoes, a high glycemic food like white rice, this is how much insulin your pancreas has to pump out to keep your blood sugars in check. But what if you added some tuna fish? Tuna's got no carbs, no sugar, no starch. Wouldn't make a difference, right? Or maybe be even lower than the mashed potato spike by lowering the glycemic load of the whole meal. But instead, you get this. Twice the insulin spike. Right? Same with white flour spaghetti and white flour spaghetti with meat. The addition of animal protein makes the pancreas work twice as hard. You can do it with straight sugar water. If you do like a glucose challenge test to test for diabetes, where you drink a certain amount of sugar, this is the kind of spike in insulin you get. But if you take in the exact same amount of sugar, but with some meat added, you get this. And the more meat you add, the worse it gets. Just adding a little meat to carbs doesn't seem to do much, but once you get up to like a third of a chicken breast worth, you can elicit a significant increased surge of insulin. This may explain why those eating plant-based diets have such low diabetes rates, because animal protein can markedly potentiate the insulin secretion triggered by carbohydrate ingestion. Oatmeal has been used for centuries as a topical soothing agent on the skin to relieve itch and irritation in dermatology. Of course, this is coming from Johnson & Johnson, that sells a brand of oatmeal lotion, but look, if it helps with dry skin or a bug bite, I can imagine having some soothing quality, but this study shocked me. There's a class of chemo drugs, like cetuximab, that causes an awful rash. I mean, it's bad enough you have some horrible cancer, but then to have some painful, itchy rash on top of it. Various treatments have been tried and failed. There's no clear preventive or curative treatment for this eruption, or is there? The researchers had heard about this study, in which human skin fragments from plastic surgery were subjected to an inflammatory chemical, and adding an oatmeal extract appeared to help. So, hey, what do you have to lose? Of the 10 patients with chemo rashes they were able to get access to try some oatmeal lotion, 6 had a complete response, and 4 a partial response, giving an overall oatmeal response rate of 100%. Doctors wrote in from around the world. Significant improvement in all patients seemed rather too good to be true, but out of desperation they tried it, and got the same astonishing results. Oatmeal, a simple topical agent producing such spectacular benefit where more complex therapies have failed. In an age when ever more expensive treatments are consistently being championed, it'd be a great pity if this inexpensive, natural approach to relieving distressing symptoms were to be overlooked. Ironically, 2 of the cancer cell lines found resistant in vitro to this kind of chemotherapy were found to be sensitive to avenanthramides, which are unique phytonutrients found in oats, suggesting that people should apply oatmeal to their insides as well. If oatmeal is so powerful that it can clear up some of the ravages of chemotherapy just applied to the skin, what might it do if we actually ate it? The pharmacology of oatmeal. Oats are reported to possess varied drug-like activities, like lowering blood cholesterol and blood sugar, boosting our immune system, anti-cancer, antioxidant, anti-atherosclerosis. In addition to being a topical anti-inflammatory, it may also be useful in controlling childhood asthma, body weight, etc. Whole grain intake in general is associated with lower risk of type 2 diabetes, cardiovascular disease, and weight gain. All the cohort studies on type 2 diabetes and heart disease show whole grain intake is associated with lower risk. They observed the same for obesity, consistently less weight gain for those who consumed a few servings of whole grains every day. Yes, all the forward-looking population studies demonstrate that higher intake of whole grains is associated with lower body mass index and body weight gain. However, these results do not clarify whether whole grain consumption is simply a marker of a healthier lifestyle, or a factor favoring per se lower body weight. For example, high whole grain consumers, those who eat whole wheat and brown rice and have oatmeal for breakfast, tend to be more physically active, smoke less, and consume more fruits, vegetables, and dietary fiber than those that instead reach for Fruit Loops. Statistically, one can control these factors effectively, comparing only non-smokers to non-smokers with similar exercise and diet, as most of the studies did, and they still found whole grains to be protective via a variety of mechanisms. So, for example, in helping with weight control, the soluble fiber of oatmeal forms a gel in the stomach, delaying stomach emptying, making one feel full for a longer period, which helps with weight loss, and then there are other effects in the small and large intestine. So it all seems plausible that whole grain intake does indeed offer direct benefits. However, only results from randomized controlled intervention studies can provide the evidence of cause and effect. In other words, the evidence is clear that oatmeal consumers have lower rates of disease. But that's not the same as proving that if we start to eat more oatmeal, our risk will drop. To know that, we need an interventional trial, ideally a blinded study where you give half the people oatmeal and the other half fake placebo oatmeal that looks and tastes like oatmeal to see if it actually works. As you can imagine, this has not been done until now. Double-blinded, randomized trial of overweight and obese men and women, and almost 90% of the real oatmeal treated subjects had reduced body weight, compared to no weight loss in the control group, a slimmer waist on average, a 20-point drop in cholesterol, and an improvement in liver function. Non-alcoholic fatty liver disease, meaning a fatty liver caused by excess food rather than excess drink, is now the most common cause of liver disease in the United States, found on autopsy in up to 90% of obese individuals, and can lead in rare cases to cirrhosis of the liver, cancer of the liver, and death. Now, theoretically, whole grains could help prevent and treat fatty liver disease. But this is the first time it had actually been put to the test like this. A follow-up study in 2014 confirmed these findings of a protective role of whole grains, but refined grains were associated with increased risk, so one would not expect to get such wonderful results with Wonder Bread. Natural immunomodulators, something that can help regulate our immune system without side effects, has been sought for centuries, and all the while, they've been sitting in the produce aisle. Plants produce thousands of active compounds, many of which modulate our immune system enough to protect us from infection. But we can't forget the fungi. Mushrooms have been used for centuries as folk remedies, and for good reason. Some have been shown to boost immune function as well. So much so, a type of fiber found in shiitake mushrooms is approved for use as adjunct chemotherapy, injected intravenously to help treat a variety of cancers by rallying our immune defenses. More than 6,000 papers have been published on these so-called beta-glucans, but almost all the data about preventing infections has come from a petri dish or lab animal studies, until a few years ago, when a series of experiments on athletes showed beneficial effects. But that was like in marathon runners. What about the rest of us? We didn't know until now. Beta-glucan fiber, found in bakers, brewers, and nutritional yeast, helps to maintain our body's defenses against pathogens even in non-athletes, according to a double-blind, randomized, placebo-controlled trial, reducing the recurrence of infections by the common cold by 25% in those that ate the equivalent of about a spoonful of nutritional yeast a day, and had fewer cold-related sleeping difficulties when they did get sick. What about a half a spoonful a day's worth? Still worked. Big drop in common cold incidence and a reduction in symptoms as well. Why, though? This study found that not only were upper respiratory infection symptoms diminished, but that mood states appeared to improve as well, like a significant boost in feelings of vigor. So they suggested, well, maybe the yeast fiber is able to counteract the negative effects of stress on the immune system. In terms of side effects, 2 folks reported stomach aches, but they were both in the placebo group. Unlike antibiotics and antivirals, which are designed to kill the pathogens directly, these yeast compounds instead appear to work by stimulating our immune defenses, and as such don't share the same antibiotic side effects. They stimulate our immune defenses presumably because our body recognizes them as foreign. But if it's treated like an invader, might it trigger an inflammatory response? Turns out, it may actually have an anti-inflammatory effect, suggesting nutritional yeast may offer the best of both worlds, boosting the infection-fighting side of the immune system while suppressing the inflammatory component. So oral intake can be considered safe and effective. Note they say oral intake, though. I would not recommend injecting nutritional yeast into your vein, no matter how much you like the stuff. Yeast is high in purine, so those with gout, uric acid, kidney stones, and new organ transplant recipients may want to keep their intake to less than a teaspoon a day. But for everyone else, is there any downside to eating nutritional yeast? Well, if you look at some packages of nutritional yeast, in California some are slapped with Prop 65 warning stickers, suggesting there's something in them exceeding cancer or birth defect safety limits. I called around to the companies, and it turns out the problem is lead. California state law says a product cannot contain more than half of a microgram of lead per daily serving. So I contacted the 6 brands I knew about and asked them how much lead was in their products. Cal originally said less than 5 parts per million, but when we called back they said less than 3 parts per million. But even if it's 3, that translates into less than 45 micrograms per serving, nearly 100 times more than the California limit. But perhaps better than Bob's Red Mill or Frontier Co-op, who evidently don't test at all. But at least they got back to me. Red Star brand failed to respond to multiple attempts to contact them. Now Food said, of course we test for lead. That's nice, and claimed that at least their recent batches meet the less than half a gram California standard. Unfortunately, despite repeated attempts, they did not provide me with documentation to substantiate what they were telling me. My favorite response was from Bragg's, who sent me the analysis certificate from the lab showing less than 0.01 parts per million, which means at most less than half the California standard, which I believe is the most stringent in the world. To put the numbers in context, in determining how much lead manufacturers can put into candy likely to be consumed frequently by small children, the Food and Drug Administration would allow like 2 micrograms a day in the form of lollipops, but look, as far as I'm concerned, the less lead, the better. Lou Gehrig's disease, known as amyotrophic lateral sclerosis, or ALS, strikes healthy middle-aged people seemingly at random, and of the major neurodegenerative diseases, it has the least hope for treatment and survival. Although mental capabilities stay intact, ALS paralyzes people, often from the outside in, and most patients die within 3 years when they can no longer breathe or swallow. At any given time, an estimated 30,000 are fighting for their life with it in this country. We each have about a 1 in 400 chance of developing this dreaded disease. So ALS is more common than generally recognized, an incidence rate now close to that of multiple sclerosis. What causes it? Well, 50 years ago, scientists found that the rate of ALS among the indigenous peoples on the island of Guam was 100 times that found in the rest of the world, potentially offering a clue into the cause of the disease. So instead of 1 in 400, in some villages in Guam, 1 in 3 adults died of the disease. Cycad trees were suspected, since the powdered seeds were a dietary staple of the natives, and there were reports of livestock showing neurological disease after eating from it. And indeed, a new neurotoxin was found in the seeds called BMAA. Maybe that's what was causing such high levels of ALS. But the amount of BMAA in the seeds people ate was so small, it was calculated that people would have to eat like 1,000 kilograms a day to get a toxic dose. That's like a ton of seeds daily. so the whole cycad theory was thrown out, and the trail went cold. But then famed neurologist Oliver Sacks and colleague had an idea. Cycad seeds were not all the natives ate. They also ate fruit bats, stewed in coconut milk. And guess what these so-called flying foxes ate? Cycad tree seeds. So maybe this is a case of biomagnification up the food chain. Remember how you'd have to eat like a ton of seeds worth of BMAA to run into problems? Well, guess how much builds up in the flesh of flying foxes? A ton worth of BMAA. And natives also ate other animals that foraged on the seeds. The final nail in the coffin was the detection of high levels of BMAA in the brains of 6 out of 6 native victims of the disease on autopsy, but not in control brains of healthy people that died. So with the final puzzle piece apparently in place, the solution to this mysterious cluster on some exotic tropical isle of ALS-PDC, so-called because the form of ALS attacking people in Guam also had signs of Parkinson's disease and dementia, so they called it ALS Parkinsonism Dementia Complex. So for the heck of it, when the researchers were choosing a comparison group of control brains, they threw in 2 cases of Alzheimer's disease. And they had BMAA in their brains too. But these were Alzheimer's victims in Canada, on the opposite side of the globe. So they ran more autopsies. No BMAA in the control brains, but BMAA was detected in all of the Canadian Alzheimer's victims tested. Wait a second. Canadians don't eat fruit bats. Well, the neurotoxin isn't made by the bat. Yeah, it's made by the trees, but Canadians don't eat cycad trees either. Their flag doesn't look like this. Turns out that cycad trees don't make the neurotoxin either. A blue-green algae that grows in the roots of cycad trees makes the BMAA that gets into the seeds, that gets into the bats, that gets into the people. And it's not just this type of blue-green algae, but nearly all types of blue-green algae found all over the world produce this neurotoxin. Up until only about a decade ago, we thought this neurotoxin was confined to this one weird tropical tree, but now we know the neurotoxin is created by algae throughout the world, from Europe to the US, Australia, the Middle East, everywhere. So if these neurotoxin-producing blue-green algae are ubiquitous throughout the world, maybe BMAA is a cause of progressive neurodegenerative diseases, including ALS, worldwide. Researchers in Miami put it to the test. Maybe the Canadians were a fluke. No. The researchers found BMAA in the brains of Floridians who died from sporadic Alzheimer's disease and ALS, but not in the brains of those died from a different neurodegenerative disease called Huntington's, which we know is caused by a genetic mutation, not some neurotoxin. Significant levels of BMAA in 49 out of 50 samples from 12 Alzheimer's patients and 13 ALS patients. The results for American Alzheimer's and ALS patients from the Atlantic Southeast compared to Canadian Alzheimer's patients from the Pacific Northwest suggest that exposure to BMAA may be widespread. The same thing was then found in the brains of those dying from Parkinson's disease. You can even pick up more BMAA in the hair of live ALS patients compared to controls. So, is BMAA, this neurotoxin, found in Florida seafood? All over the place, in freshwater fish and shellfish like oysters and bass, and out in the bay. In fact, some of the fish, shrimp, and crabs had levels of BMAA comparable to those found in the fruit bats in Guam. In the US, the fish may be the fruit bats. These data on BMAA neurotoxin concentrations in animals in South Florida waters indicate that the situation in Guam is not unique. It looks like BMAA could be found in high concentrations in aquatic animals in many areas of the world. This could explain ALS clustering around lakes in New Hampshire, up to 25 times the expected rate of ALS, with some families eating fish several times a week. or in Wisconsin, where the most significant ALS risk factor was the past consumption of fish out of Lake Michigan, or clustering in Finland's Lakeland district, or maybe seafood eaters in France or around the Baltic Sea building up, particularly in fish, mussels, and oysters. When I think algae blooms, I think the Chesapeake Bay, near where I live, that gets choked off thanks in part to the poultry industry pollution. And indeed, there was a recent report linking BMAA exposure to ALS in Maryland. ALS victims living within a half-mile of each other raised some eyebrows at the Hopkins ALS Center. The victims each ate Chesapeake Bay blue crabs every week, and so they tested a few, and 2 out of the 3 crabs tested positive for BMAA, indicating that the neurotoxin is present in the aquatic food chain of the Chesapeake Bay and a potential route for human exposure. To bring the story full circle, things in Guam are looking up. The ALS epidemic there may have been triggered by their acquisition of guns, but now the epidemic appears to be over thanks to the near extinction of fruit bats due to overhunting. But while the rates decline in Guam, neurodegenerative diseases like ALS around the rest of the world are on the rise. It's plausible that humans have been exposed to some level of BMAA through our evolutionary history, but the increase in algae blooms as a result of human activities is probably increasing this exposure. There's a general consensus that harmful algal blooms are increasing worldwide thanks in part to industrialized agriculture. More people means more sewage, fertilizer, manure, which may mean more algae, which may mean more exposure to this neurotoxin, leading to a possible increased incidence of neurodegenerative diseases such as Alzheimer's, Parkinson's, and ALS. BMAA is a strong contender as the cause of, or at least a major contributor to the cause of both endemic and sporadic ALS and Alzheimer's disease, and possibly conferring risk for Parkinson's disease as well. The ramifications of this discovery are enormous. With substantial and ever-growing evidence that BMAA does play a role in the onset and progression of neurodegenerative diseases, the most important question is, what mode of activity does BMAA exert? What? No, it's not. The most important question is, how do we reduce our risk? We know that the presence of BMAA in aquatic food chains could be a significant human health hazard. There may even be a synergistic toxicity between mercury and BMAA, making certain fish even riskier. Until more is known about the possible link of BMAA to Alzheimer's and ALS, it may be prudent to limit exposure of BMAA in the human diet. Previously, I explored how adding berries to our meals can actually blunt the detrimental effects of high glycemic foods. But how many berries? The purpose of this study was to determine the minimum level of blueberry consumption at which a consumer may realistically expect to receive antioxidant benefits after eating blueberries with a sugary breakfast cereal. If we eat a bowl of cornflakes with no berries, within 2 hours, so many free radicals are created, it puts us into oxidative debt. The antioxidant power of our bloodstream drops below where we started from before breakfast, as the antioxidants in our bodies get used up. And a quarter cup of blueberries didn't seem to help much, but a half a cup of blueberries did. What about fruit for diabetics? Most guidelines recommend eating a diet high intake of fiber-rich foods, including fruit, because they're so healthy. Antioxidants, anti-inflammatory, improve artery function, reduce cancer risk. However, some health professionals have concerns about the sugar content of fruit, and therefore recommend restricting fruit intake. OK, let's put it to the test. Diabetics were randomized into 2 groups, one told to eat at least 2 pieces of fruit a day, and the other told to at most eat 2 fruits a day. The reduce fruit group reduced their fruit, and it had no effect on the control of their diabetes or weight, and so the intake of fruit should not be restricted in patients with type 2 diabetes. An emerging literature has shown that low-dose fructose may actually benefit blood sugar control, so having a piece of fruit with each meal would be expected to lower, not raise, the blood sugar response. The threshold for toxicity of fructose may be around 50 grams. The problem is that's the current average adult fructose consumption, thanks to industrial sugar intake, table sugar, and high fructose corn syrup. So the levels of half of all adults are likely above the threshold for fructose toxicity, and adolescents currently average 75. Now, is that the limit for added sugars or for all fructose? If we don't want more than 50, and there's like 10 in a piece of fruit, should we not eat more than 10 fruit a day? Quoting from the Harvard Health Letter, "The nutritional problems of fructose and sugar come when they are added to foods. Fruit, on the other hand, is beneficial in almost any amount." What do they mean, almost? Can we eat 10 fruit a day? How about 20 fruit a day? It's actually been put to the test. 17 people were made to eat 20 servings a day of fruit. Despite the extraordinarily high fructose content of this diet, presumably about 200 grams a day, 8 cans of soda worth, the investigators reported no adverse effects, and possible benefit actually, for body weight, blood pressure, and insulin and lipid levels after 3 to 6 months. More recently, Jenkins and colleagues put people on about a 20 servings of fruit a day diet for a few weeks, and no adverse effects on weight or blood pressure or triglycerides, and an astounding 38-point drop in LDL cholesterol. There was one side effect, though. Given the 44 servings of vegetables they had on top of all that fruit, they recorded the largest bowel movements, apparently, ever documented in a dietary intervention.